much. I think we've already heard at this meeting how diverse RNA is and how fun it is to work on. And we heard a little bit yesterday about RNA as a kind of gene regulatory molecule. And so that's what I'm going to focus on today. And if you are an aspiring gene regulatory molecule, one nice feature to have is to be able to locate yourself to the right place at the right time. And RNA does that, in some cases through direct interactions with the DNA or with the chromatin. And that determines a genome-wide RNA-DNA kind of localization to allow RNA to make those regulatory changes at times when the cell needs it. And so how do we go around and study RNA localization on the genome? In previous years, a lot of work is focused on these kind of one-to-all methods, which you can think of analogously to kind of chip sequencing for transcription factors. And so that applies in cases where you have a single RNA that you're interested in. You cross-link that, similarly to how we heard for cross-linking to proteins. And then using kind of biotinylated antisense probes against your RNA, you can pull down the regions of the DNA where it was bound and call peaks, etc. That has quite a clear drawback in that it only provides you information with one RNA at a time. And as we've heard, there's such an abundance of RNAs around that the field has switched predominantly now to kind of all-to-all methods. And we heard a little bit about these yesterday afternoon from Lena Ma's talk. And these include methods like radical sequencing and grid sequencing. And what this relies on is that you have many RNAs that you're interested in. Again, you cross-link those to the DNA sites that they're in localization with. And then instead of using antisense oligos against the RNA itself, you use biotinylated linker oligonucleotides, which link together a free end of the RNA with a free end of the DNA, and creates a kind of chimeric sequencing read where one end of that read encodes information on the RNA that was there, and the other end, the region of the DNA. And so our kind of functional unit that we're working with is, as I said, we have a chimeric sequencing read. We have the DNA read, or the DNA end of the read, which we can align to the genome and summarize by bins of the genome. And the RNA end of the read we can take and align again to the genome or transcriptome and then summarize that by genes. And so what we end up with is an interaction between an RNA and a bin. And we can summarize that essentially by counting these reads across millions and millions of sequencing reads. We can count up how many reads support that interaction between the RNA and the bin. And in an ideal world, we would just count those up, call the sort of top performing ones, go to the pub and publish the paper. But unfortunately, we started looking into these a bit more, and what we saw first is that these ligation read counts correlate really strongly with the abundance of the RNA, and that's pretty expected, right? I mean, in these two cases, in this case, we have RED-C, which is this all-to-all RNA-DNA sequencing method. And then if we compare that to RNA sequencing abundance of the same RNAs, we see a really nice correlation. But what this started to suggest is that maybe these methods aren't just capturing, the gene regulatory interactions that we're really interested in, but maybe they're also capturing, a bit of nascent transcription. And so we started to look where these reads fall. And so we looked at TEAD1, which was the highest expressed RNA in the data set that we were considering at this point. And we looked at where its reads were falling. And so at 0 megabases here is the locus of TEAD1. And you can see that the vast majority of these reads, in 40 megabases of space, fall at the locus, which isn't great. It kind of shows, and we have other data to suggest this, is that this is nascent transcription and potentially not kind of bona fide RNA-DNA interactions. And it's not just the case for mRNAs. We actually looked at MALAT1, which is famously chromatin localized, and we see a very similar signal, which was a bit disappointing. And this is prevalent throughout these data sets across all of the different methods. And this really suggests that if we want to call true RNA-DNA interactions from these data, we have to come up with a background model that takes into account this prevalence of nascent transcription. And so that's what we did. We came up with a tool that we've called RADIANT, and RADIANT stands for RNA and DNA interactions analyzed and tested. And what that does is essentially what I've just been speaking about. We take every RNA that's in the data set, we look in a window around the locus of the RNA, and we look at the relative interaction frequency of that RNA at given distances from its own locus. And we do that across all the RNAs in the data set, and if we take a mean of those values, we can come up with quite a nice background distribution that you can see would decay as you go further and further away from the RNA's own locus. Obviously, there are many sites in the genome which aren't in proximity to the RNA's locus, so how do we deal with those? We look genome-wide at the bins that we have, we look at RNA interaction frequencies per bin in the genome. And by taking a mean of those, we actually come up with a mean expected interaction frequency per bin of the genome. And so what we can do then is we can combine these two backgrounds. So if we have our RNA that we're interested in, we can have the background based on its transcription start site, essentially, alongside the biases that might be present for bins as well. And what we do then in quite a stringent way is we take the maximum background value at each bin in the genome relative to the position of the RNA we're interested in. And what this allows us to do is then call RNA-DNA interactions against that background. And we do that using a Poisson test by taking the observed counts that we see for the interaction versus the expected counts that we can derive from that background. And so what does this look like in practice? What happens when we really start to call RNA-DNA interactions? This is a fairly busy slide, so I'll walk you through it. What we're looking at here is the MALAT1 gene locus in mouse across 3 different all-to-all RNA-DNA sequencing methods. So we have a grid sequencing, and we have 2 separate radical sequencing experiments. And right here in the middle of the plot is the MALAT1 gene locus. And what you see in white for each of the methods is the background we build for that data. So you can see it decays as we go further and further away from the locus. And in blue, the kind of spiky bits, that's the MALAT1 DNA interaction frequency at that location. And where there's a little black dot above the spike, that means we call some kind of significant interaction, and that scales in size with how significant it was. And what this does is it shows that the background is doing its job, even though you can see the interaction frequencies are objectively higher. Closer to the locus, we actually call fewer and less significant interactions there compared to this space at 9 megabases. And actually, we call that region very, very consistently across these 3 different data sets, implying that Radiant can consistently identify bona fide RNA-DNA interactions independent of the input data. And so that's all very well and good. That's for one RNA, that's in cis. You might be asking, there are all these other RNAs present in the genome. But actually, when we correlate the significance of the interactions that we call, and this is looking at all interactions now, we can see that the p-values we assign to the interactions across grid, radical, they look quite consistent. So we can call consistent RNA-DNA interactions in a genome-wide manner across all of the RNAs that are present. So consistent interactions are all well and good, but are these interactions really relevant? Are they really gene regulatory? And so we set about trying to evaluate the performance of Radiant relative to those one-to-all sequencing methods that I described to you at the start of the talk. And so we came up with a scheme where we would take our genome and we would bin it. We would then use orthogonal one-to-all RNA-DNA interaction data to label those bins. So something like wrap or chart or chop. We would then look at how our RNA-DNA interaction statistic looks at that bin for the RNA that matches the RNA in the one-to-all data. And we can use that to evaluate the performance in essentially a classification-based manner. So we did that across several data sets. We had two sets of that for mouse, so both looking at MALAT1, but with different all-to-all sequencing data. And we also had it in human, but then that was different one-to-all data. So we had a one-to-all data set for MALAT1 and a one-to-all data set for NE1. And then we looked at different interaction callers. So we looked at Radiant, which I've described to you. We used BARDIC, which utilizes trans mRNA contacts around the genome, as well as an element of distance, and then 2 other RNA-DNA interaction callers as well. And the good news for us was that when we looked at the results, so you can see here area under the precision recall curve and area under the ROC curve, Radiant consistently outperformed these other interaction callers. And so we thought, great, project over, let's move on. But actually, if you look a bit closer at these y-axis values, you'll notice they're not necessarily particularly impressive. You've got kind of values of 0.6 something here, maybe creeping up towards 0.7 in area under the ROC curve. And you and the reviewers can very validly ask, why is the performance of these interaction callers so poor? And we also, wondered that ourselves. And actually, we started thinking, are we really using the right evaluation process to do this? We're essentially using these interactions to try and classify whether we're true or false. Maybe we should look in a more correlation-based manner, because that's actually more what we're doing. And so we switched tracks a bit, and we started to think about how we could use GSEA, so gene set enrichment analysis, to try and statistically associate the statistics coming out of these interaction callers with the presence of orthogonal peaks. And so the way GSEA works, I might be preaching to the choir, but allow me. So what we do is we rank every bin in the genome by our RNA-DNA interaction statistic. We then look at how often those bins contain a peak from the orthogonal data. And what it does is it calculates a running sum as it walks down that list of rankings and adds the enrichment score. And so you can see for ranking 1 here, this would result in quite a nice enrichment score for that because the peak bins are located towards the top of the ranking set. For ranking 2, you can see these are kind of more randomly distributed throughout the list, and this would produce then a lower enrichment score relative to ranking 1. And so we did this for each of our interaction callers and each of our interaction data sets. And this is an example. So this is neat1 one-to-all data compared to grid sequencing. And what we could see is that we saw a much higher enrichment score for radiant as compared to these other RNA-DNA interaction callers. And that wasn't just present in that one example, that was present across essentially all of the examples that we looked at. And so when we combine this classification-based approach and the correlation-based approach, we can see that radiant handily outperforms these other methods, although it must be stated. In some cases, it is rather close. And so we can say radiant seems to accurately identify RNA-DNA interactions in a genome-wide manner. That's useless if you need all the compute in the world to be able to run these analyses. Actually, what we figured out is that radiant is fairly computationally efficient. So particularly compared to Bardic, which was the closest sort of competition in terms of performance, you can see that actually you can run radiant interaction calling on a local machine relatively quickly. So it's fairly lightweight as well as being reasonably effective. And what's not shown here is that we've integrated this interaction calling into a SnakeMate pipeline, which really takes you all the way from the raw sequencing reads for these methods, all the way through to the called interactions. You might just have to ask a friend who has a bit more RAM to do all the alignments and things like that. And so that's great. We've got a consistent and accurate RNA-DNA interaction caller. But a big theme of this meeting has felt like, we come up with these tools, but the real science is then applying those tools to learn something new about biology. And so we wanted to look for true gene regulatory RNAs and non-coding RNAs. And so what we did is we utilized radiant in a scenario of endothelial to mesenchymal transition. And you'll have to forgive me because I come from the cardiovascular field, but you may not be familiar with this term, but it's essentially a phenotypic switch between endothelial cells into a more mesenchymal-like cell, which proliferates more and is a bit more inflammatory. And this is a phenotypic switch that we see in several cardiovascular diseases like atherosclerosis or myocardial infarction. And you can reproduce this switch experimentally quite nicely. And it gives us this really nice kind of dynamic process to study things like RNA-DNA interactions in. And so we did this experiment. We generated nuclear RNA sequencing and radical sequencing across the control and the treated cells. And what you can see here on the right-hand side of the slide is that we could identify an awful lot of long non-coding RNAs who were dynamic not only in their expression on the x-axis, but also in their association with chromatin on the y-axis. Not always, both, sometimes one or the other, but equally very interesting. And so we wanted to validate whether some of these link RNAs are true gene regulatory factors. And so we selected one of them, which was this link RNA called NUTN2B antisense 1. I'm just going to call it NUT from this point forward. And we essentially reproduced its downregulation in endo-MT by knocking it down with siRNA. And we then looked at differentially expressed genes, as you would, perfectly standard. But what we did is we looked at those differentially expressed genes and asked the question, was there a NUT binding site at that gene locus that we saw in the radical sequencing? And all the genes that you see highlighted in this volcano plot, we saw a NUT binding site across the locus of that gene. And that was actually statistically significant in terms of there being an enrichment for NUT binding sites at genes which were differentially expressed following depletion of NUT by siRNA. And we saw this in a phenotype which is important to endothelial cells in the cardiovascular system. So we then asked, are these genes which are putatively regulated by NUT important for endothelial cell behavior? And so actually some of the pathways that pop up look very promising, things like extracellular matrix interaction, cell junction organization, and cell cycle. Obviously, all very important for endothelial function. So the question then becomes, okay, do we see these putative NUT-regulated genes in ENDO-MT in that original scenario that we looked at the data in? And so what we did is we went back and we took a single cell data set of ENDO-MT. And what we did is we then looked at theoretically NUT down-regulated genes or NUT up-regulated genes. And we looked at whether these gene modules behave in the way we would have expected with the loss of NUT expression during ENDO-MT. And what we see is that it seems to map. So genes that we would expect to be down-regulated following the down-regulation of NUT behave in exactly the way they should do in the single-cell RNA dataset. And correspondingly, genes that we would expect to be up-regulated show the same behavior. And so this implies that this RNA, NUT-M2B antisense 1, seems to have potential to be actually a gene regulatory link RNA, although mechanistically how it exactly does that requires further evaluation. And so I've shown you that we're accurate, we're consistent, we're efficient. It's all wrapped up in a nice SnakeMate pipeline ready for people to use. And hopefully, we can really identify gene regulatory non-coding RNAs using this tool. So I'll go through that quickly. And the only thing, obviously, to acknowledge is that many people were responsible for this work, notably Simone Desire, who's my PhD student, and she did all of the dirty work that's behind this project. She made the wise decision to go on vacation. And so, unfortunately, you're stuck with me presenting this work instead of her. Also, Ralph Brandes, my supervisor, or my boss in Frankfurt, and Marcel Schultz, whose advice is really invaluable for this project. And if you'd like to use Radiant, it's available on GitHub. If you'd like to read the preprint, it's on bioRxiv. And I'd like to thank all of the funders that made this research possible. And I'm happy to try and answer any of your questions if you have them.
Thank you. So any question for Tim?
Thank you for the great talk. I just was wondering what is the half-life of the RNAs that exert this regulatory function? And also I'm wondering if these methods that detect the location of the RNA, they could mistake. And let's say we have some intragenic enhancer that is within one of these genes, and we are targeting the gene, but indeed what we are targeting is the enhancer within the gene. That is what is executing the regulatory function. Thank you.
Yeah, so I'll answer the first question first, as well as I can. It's a really interesting question, right? How long is a potential RNA that's in the nucleus active for? How long can it be there and work? The answer is we don't really know. I guess the only way to do that would be to conduct these methods in a manner similar to something like SLAM-seq, where you can metabolically label the RNA and see how long it sticks around for. Obviously, in the past couple of years, we've had these crazy examples of long-lived RNAs. So, RNA has the potential to be long-lived and act. But unfortunately, I can't give a concrete answer. Your second point is also very interesting. So in terms of, it's a little bit chicken and egg, right, actually, because unless I'm getting your question wrong, you're saying, what if these interactions are essentially consequences of the genome conformation coming together? Or did I misinterpret that? Sorry.
As far as I understood the method detects where this RNA is binding. And I just was thinking that some genes, they have intragenic enhancers, might have some RNA product. And instead of, we think that it is this transcript, what is in this location of the genome, but it is in this enhancer with the gene that is in this region of the genome.
Yeah, that could very well be. I mean, I can't pretend that these methods are perfect. They're really not. We see massive attrition. So in terms of the sequencing data you get, if you go and sequence 300 million reads, you'll get 10 million reads that are actually useful in the end. So we can't be concrete on what we're really covering. The impact of 3D genome conformation on these data is also probably massive, because actually, although we show all these results in a linear fashion, the genome's not linear, right? And so actually, I think what we're seeing a lot of is that 3D genome conformation determines partly these interactions. And so that MALAT1 example I showed is probably a great example of, it's not MALAT1 diffusing for megabases across the genome and reaching this point. It's the fact the genome is in conformation with one another, and MALAT1 is just taking that little hop across.
Yeah, any other question?
Hi, team. Very nice work. I just have a question regarding how to determine it is a regulatory non-coding RNA or not. I noticed that you performed knockdown experiments and then examine the differentially expressed genes. So does it depend on the number of the differentially expressed genes after knockdown?
Yeah, it's an interesting point. So I've only showed you the nicest example we have. We actually did this for more lincRNAs. We do see similar patterns in that they tend to be enriched at genes which are differential following their knockdown. However, essentially, we always have to remember that what we're showing are independent observations, right? We're showing lincRNAs bind in certain places, and if you knock down the lincRNA, then those genes are changed. You're not linking those things together experimentally unless you start doing much more complex experiments like figuring out the region of the RNA that's important for that interaction and mutating it. Obviously, knockdowns are messy, and establishing what's a primary or a secondary effect is very difficult. So the best we can do at the moment is what I've showed you is statistical association. But in terms of really primary data showing that a lincRNA is a gene regulatory factor, it needs some smarter experiments by people who know what they're doing, I would say.
Thank you.
Okay, if there's no question, thank you again to team. Let's thank again all